Hey class, thanks for coming in, sitting down, taking a quiz quietly, and being prepared. As you know, I've been talking about Mendel and his contributions to genetics, as well as genetics in general. So here's our past vocab. These are all terms you guys should already know and have studied since our last meeting time. But let's go ahead and get started. We're going to talk about heredity today. What do these two pictures have to do with genetics? Here's another question. Why don't all these puppies look the same? Or why don't all these kittens look the same? They have the same mom. They have the same dad. Why don't they all look the same? Why do you, as an individual, not look exactly like your mom or exactly like your dad? Something to think about as we go through today's lesson. So in those pictures, what were some characteristics that you saw that were similar between the kittens compared to kittens, puppies compared to puppies? What were some of the similarities you saw? There were color similarities between some of them. There were fur patterns that were similar. They were about the same size, same shape. So what if I were to tell you that all of these characteristics can be contributed to genetics? And they're actually controlled by genetics. We're going to talk about the way traits are transferred from one generation to the next. So a generation, can anybody tell me what a generation is? Okay, so a generation is all the offspring that are the same stage of descent from one common ancestor. So y'all have grandparents, and your grandparents had kids. So your grandparents are a generation, your parents are a generation, and you guys are another generation because you are offspring of your parents. So you guys are a generation. So we are able to predict the heredity of traits from generation to generation using a Punnett square. Now, on your quiz I asked you what a Punnett square was, and I'm sure some of you were just like, Mrs. Sorensen, we have no idea what a Punnett square is. Are you crazy? So, here's what a Punnett square is. It's simply a diagram that shows the predictions by considering all possible gamete combinations. Gamete over here. So this is the simplest Punnett square. 4 squares inside of another square. And what you'll get in here is a product of possible gamete combinations. And then on the outside, you'll have your possible gametes. So also on your quiz, I wanted you to do a cross between big Y, and little y, little y. Once again, you're probably like, what are you talking about? Well, this here is called a monohybrid cross. And what a monohybrid cross is a cross involving one pair of contrasting traits. And this allows us to predict the expected results of genotypes and phenotypes. And this is very important for plant and animal breeders because then they can breed for desirable traits and try to avoid undesirable traits. So this information for the monohybrid cross can be put into a Punnett square. So let's go ahead and do that. Big Y. So you're taking one allele per each square. So you've got big Y, little y, big Y, little y, big Y, little y, big Y, little y. Okay? So you're just pairing these squares. Pairing these squares to get your genetic combinations or your gamete combinations. So we talked about Mendel, right? And one trait that he was interested in with his pea plants was seed color. He found that the dominant seed color was yellow, and the recessive seed color was green. So you have a homozygous dominant, homozygous recessive. Homozygous dominant gives you yellow, homozygous recessive gives you green. So what do you get when you have a heterozygous? Remember we talked about dominant alleles covering up recessive alleles, and coming out with a phenotype of a dominant. So this guy will give you yellow too. Yellow seeds. So here are possum combinations. So what do we have here? We've got 4 heterozygous plants, and we have 4 plants that give you what color seeds? Yellow. So you have 4 yellows. Okay, these 4 yellows. This is the P generation or the parental generation. So if you wanted to create some more offspring from this parent generation, you take these offspring, so let's say we'll do these 2, and put them into the F1, which we talked about in class last time. And what goes here? Big Y, little y. Big Y, little y. And this will give you the new gamete combinations. So what do we get here? How many yellows do we get? We get 3 yellows and 1 green. Or you can look at it as a percentage. 75% yellow, 25% green. So you get 3 out of 4 and 1 out of 4. All different ways of looking at it. And we'll come back to that in a minute. So, let's consider a situation where Mendel was given a seed. And he was given a yellow seed. And he had a friend who wanted to know its genotype. So Mendel's given a seed. He doesn't know what it is. So if it's a yellow seed, what are the possible genotypes? You've got big Y, and then big Y, little y, correct? And what the purpose of it, at this point you would perform a test cross. The purpose of a test cross is to discover which genotype it actually is. So you take the unknown and you cross it with a homozygous recessive, which is your YY. Now you'll end up with one of 2 outcomes. So here you have your big Y, and you don't know what it is. Big Y, don't know what it is. We have to do big Y, right? And then you do them both against homozygous recessives. Now, Punnett squares are not something you do to discover this. A test cross is actually breeding those plants together to come up with a final product. So you get a YY, YY. So if this unknown is a big Y, here are your combinations. If your unknown is big Y, little y, here are what your offspring will yield. Okay, so here, what do you have? How many yellows? How many dominant? Okay, you have 4 yellows. Or you have 4 out of 4 yellows. Or you have 100% yellow. Here, how many yellows do you have? 2. So you have 2 yellows. Or you have 2 out of 4 yellows, which is also 1/2. Or you have 50% yellow. True? All right, so if it's a homozygous, dominant, unknown, then you'll have 100% yellow offspring. If it's heterozygous, then you just get 50% yellow. Makes sense? All right, so here we are talking about probability. So this is showing us the likelihood of that a specific event will occur. So a likelihood of getting yellows. The likelihood of getting yellows in this one, if you're crossing these gametes. So in order to get this probability, you take the number of your outcome, so the number of yellows, and you put it over the total available. So in this case, with a simple Punnett square, the number is always going to be over 4. So as we're closing, just a teaser for tomorrow, we're going to go ahead and take a look at some human dominant and recessive traits and figure some probabilities within our class. Okay, go ahead and start working on your worksheets. I will be handing out. You'll have 2 of them. If you finish the one, go ahead and move to the other. If not, we've got some other instructions as we move into that. Okay, thank you.